Okay, I'm going to start off by just apologizing, because I find that people look at this bio-nano stuff as really, really spooky and scary. And so I'm just going to say in advance, I'm sorry, that's my Canadian side. You're not going to like all of this, but it will be a great conversation starter moving forward. The reason why so much of this is spooky is because it is unfamiliar to us. Now, I want to give you a little taste of why it's unfamiliar. We've always had the stars. We've always been able to see it as long as we've been human beings. But looking down is really new. It's only about 400 years old that we started to make microscopes. Before that, we had absolutely no idea there were things smaller than the naked eye could see. And trust me, it is a crazy world. This is just a couple of drops of pond water under a microscope. There are creatures there that you can barely imagine. The oceans are filled with these creatures. It is truly an alien world, the microscopic. Now, just to give you a sense of scale, these are things you can see with your eye. I love this visualization because you start to move in. You start to see, quickly go beyond the resolution of the human eye and start to go into the bigger cells like the amoeba, which you might remember from biology, the paramecium, some of the larger cells like the human egg. Then you start moving into the smaller scales. This is a micron scale. This is bacteria, mitochondria that powers our cells. And now you start moving into the nanoscale, the viruses, and ultimately the subcellular components that make up all living systems. This is an alien world with alien physics. We're not used to seeing it in our everyday lives. Why look at small things? We build some of the biggest machines in the world to look at the smallest particles because they teach us about the universe that we live in. We study the elements to give us a foundation in chemistry and geology that helps us better understand the solar systems and the universe that we live in, our local universe. When you start moving into the micron scale and the nanoscale, you start to be able to work with new domains and physics that just don't really apply at any other scale. This stuff is almost magic. And I just want to point out, here are some nanoparticles that we've been able to engineer over the last few years. This is really the part about nanotechnology that I'll go into, and that's about it. But I also want to point out that these nanoscale particles between 1 and 100 nanometers is also where most of the subcellular components in living systems also fit. Cells are much larger. You can see they're on the micron scale, but the components of cells are down at the same size as some of these nanoparticles. We study these because they teach us about this, the living world that we inhabit. This is a living world, and it's all powered by these systems. So when you see an astronaut or someone looking into a microscope, they're really doing the same thing. They're exploring new frontiers. And granted, being an astronaut is a lot more exciting, and that's because rockets occasionally blow up. That doesn't happen so much in biology labs. We hope. But it is really an act of exploration, and we are very early in these explorations, both to space and into studying the small. There's a lot of value to it because today, as you know, all of our computers are powered by nanotechnology. The features on a semiconductor chip now are 7 nanometers. That's about 3 DNA strands stacked side by side. It's hard to imagine the scale of that. But the folks working in the semiconductor industry have been working at this level for over a decade. We're starting to see nanoparticles come into play with things like quantum dots, which were fascinating little particles that today are being used to make televisions, as well as other types of diagnostics. In fact, nanotechnology is pretty much pervasive in the world. Germany is a leader in it, according to the briefing documents that I had. I am not an expert, so I won't try and elaborate on that, but just know that the nanoscale has always been important. Biologically, nanoscale structures have been fascinating for a long time too. A gecko's foot has nanostructures that allow it to stick to almost any surface through hydrogen bonding. How does it make that? And how does a living cell operate? These are the most amazing machines to me because 1 makes 2, 2 make 4, and it does this all using elemental materials. Elemental. This is nanotechnology in action, doing things that our nanotechnologies, the one we've been building, can only dream of. And this has been happening for billions of years on this planet. Now we know how it works, sort of. This is basic biology. You take a human, you pluck out a cell, you pull out its genome, there's its chromosomes, you unravel the chromosomes, you get a long string of DNA. DNA is transcribed into RNA, the working copy of the nucleic acids, which ultimately are translated into proteins, which build us and every living thing on this planet. Biology 101 in 2 seconds. This is the reason why I love biology, though. Biology is self-assembling nanotechnology with a built-in programming language. No other nanotechnology has this. This is really important. Now, we didn't know much about this code for a long time. I remember before, in the early 1980s, just to read a few hundred bases of genetic code was enough to get your PhD. But by 1990, the Human Genome Project had officially started up after about 5 or 6 years of behind-the-scenes wrangling and getting money and politics. And this project catalyzed an amazing shift in genomics that continues to this day. It went from doing DNA sequencing like this, using X-ray film and radioactive isotopes and reading DNA code by hand, to factories that churn out DNA 24/7 using robotic sequencers like these HiSeqs made by Illumina. A couple of weeks ago, I spoke with the CEO of Illumina, and I said, give me a little heads up on what's coming. And he said, well, we've got our new NovaSeq sequencers coming out. This is essentially a smaller, faster version of the current sequencers that he claims will be able to do a human genome for $100. You saw today, or you heard earlier today, this incredible advancement in DNA sequencing technology, going from billions of dollars to now hundreds of dollars to do a human genome. That's great, but it also opens up reading the code of all living things. This isn't just about humans. And the technology continues to advance. About $500 million worth of investment went into making this little device, the Oxford Nanopore MinION sequencer. This is actually a hybrid chip. It's basically a cyborg. It's part biological, part semiconductor. And it works simply by having a pore that allows DNA to go through and you read the electrical signals right off of it. It costs $1,000 to buy this device, and you throw away the chip inside and replace it. This allows you to do DNA sequencing in the field. It also allows you to do it in space. Just last year in the summer was the first time DNA had ever been sequenced on the space station. We're still learning how to unlock the secrets of life through DNA code with our technologies. This is just the start. And as you heard earlier today, now we're able to take a drop of blood and we're able not only to get a full genome, we're also able to look for mutations, cancer cells, other infections, parasites, just from a single drop of blood. Now remember, every baby that's born gets a heel prick and is tested for metabolic errors that could essentially kill them. We've been doing this in North America, I'm sure it's done here, for over 60 years. Every baby gets a drop of blood. How much longer will it be until we do a full genome sequencing on every baby? DNA diagnostics are changing everything about medicine. This company, which was essentially spun out of Illumina, the sequencing company, has raised, it's only 2 years old, it's raised over $1 billion in financing, and just a couple of weeks ago, they announced a 120,000 person multi-center clinical trial to look for cancer markers in blood. That's remarkable. Companies doing clinical trials larger than any medical center could ever do. And this is just the start. Why is this important? Well, it's because we can't scale these things. An MRI is great for finding cancer once it's a certain size, a few millimeters. But you can find a few hundred cells that have gone cancerous with DNA sequencing technology. We've just started to tap into the power of this tool. How do we integrate all that? One of the companies at the forefront of this space, Health Nucleus, is starting to build avatars, starting to collect this data and build a digital representation of the human being. A full MRI scan, microbiome scan, genome scan, consult. And apparently about 20% of the people that go through this process, which is very expensive, $25,000 to $50,000 US dollars, about 20% of the people that go through it find some actionable medical need. How quickly can we make this type of technology cheap and available for everyone? Well, another company that I've been watching in China is trying to do that. iCarbonX is founded by the former head of BGI, the Beijing Genomics Institute, that is really the world's sequencing powerhouse. So we can already see the convergence of DNA sequencing technology and artificial intelligence for analysis at a price point that's going to be very different than Health Nucleus. But I don't think that's the end of it either. In fact, a couple weeks ago, I made a bet with this fellow. This is Gary Wolf. He's one of the founders of something called the Quantified Self Movement. People interested in measuring different aspects of themselves. And I made a bet, $1,000, that within 5 years, there will be a company that will offer free genome sequencing to everyone and will have at least 1 million members in their databases. Now, if I said to you, I will sequence your genome for free and give you all of this information for free and control over your data, how many of you would get your genome sequenced right now? About 45%, maybe 40. Now it's different. If it's with a younger audience, it's 85%. The older the audience, the fewer people are comfortable with this technology. And that's because it is one of the most powerful data sets. But today it's fragmented. You're not even allowed to access it on your own in many states in the United States. I don't know how it is in your country. Even now, if you want to get your DNA sequence, you often have to go through a doctor. This is your code. I tried to have my daughter sequenced. She's 2 years old. I can't get it done. Here is the most powerful predictive test in medicine, and I cannot get my daughter sequenced in the United States. This needs to change. Gary said, no way there will be a company that does this in 5 years, but I'm almost certain to win, because if one doesn't appear, screw it, I'll go make it. Right now, the problem is trying to aggregate all this data and make sense of it. This is a 5-year-old model of a bacterium called Mycoplasma. It was made by researchers at Stanford University, and when it was published 5 years ago, it was described as audacious because it collected all of this information about the bacterium's metabolism and put it into a MATLAB model and did some visualizations to kind of predict its metabolism. This is still the most sophisticated model of a living organism we have to date that I can show you. And it's pretty crude, because this is what your kids are seeing as models with their video games running on PlayStations or Xboxes. These are completely simulated environments. Why can't we make a simulated cell that has this type of resolution and interaction and engagement? We need to fundamentally rethink the way we're doing cell biology. But that's reading and analysis of this information. The really fun part, and this is where you might find it spooky, is when you start actually writing genetic code. This is not genetic engineering as you've thought about it in the past. This is more like software engineering. This is when you sit down and you go, I want to make something. Here's my design. How do I go and put it together using this incredible new tool of life and the code that powers it all? And by the way, if you guys work with computers, you know how weird it is, that how many languages you need to know, how many protocols you need to have, how many libraries you need to install. With life, there is one code, and it's pretty much universal from bacteria all the way to us and everything in between. And once you get adept at it, it's really simple to figure out. Let's just say that AI is going to make a massive difference here. Now, in the last decade or so, we've created a new branch to the tree of life called Synthetica. It's a horrible name. Let's just call it intentional design. And we only have 3 basic tools to start working in this medium. One of them is evolution. Pretty straightforward. The second one is editing, making small changes to a large existing genome. And the third one is actual design, when you start from scratch and you build from the bottom up. Now, I want to play this little video for you. It's the only one I have with sound, but this describes evolution because most people have no clue how quickly it can work in these organisms.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Isn't that remarkable? In just 11 days, you can evolve to use essentially an antibiotic as a food source. Now, these are very fast-growing organisms, but the principle works the same for all life. It's incredibly flexible and adaptive. And in fact, the process of evolution can be accelerated by using technology such as MAGE, which essentially automates and drives the process of evolution in an automated way. This is, again, from the Wyss Institute. But you, guaranteed, if you don't know how to design something in life, you can drive it to a certain target. We're going to be doing this all over the world as we start to need organisms to meet new human requirements. Jumping ahead to editing, you've probably heard a lot of about CRISPR in the last 5 years. This is a tool that just popped up out of nowhere and allows us to do gene editing, genome editing, really fast, really cheap. It's basically copy-paste for DNA. Luhan Yang at eGenesis is one of the leaders in this field. She's been editing the pig genome to remove endogenous viruses and to humanize the pig so that one day we can have an unlimited supply of organs. They raised $38 million recently. They're moving ahead very quickly with this work. But CRISPR has also been used to selectively kill cancer cells. There's a company in France I like called Eligo Biosciences that uses it to kill microbes. And we've also started the first human therapies with this technology. But it's when you get to design that things get really interesting. This is when you really start building intentional creatures. Today we have companies like Twist, based out of the San Francisco area. They print DNA. They essentially have a 3D printer for the DNA molecule. So if you can design the genetic code, you can just print it and then install it into a living organism. The master of this is Craig Venter. He's been at the forefront of this field for longer than I can count. Last year, he published the design and synthesis of a minimal bacterial genome. This is a synthetic bacterial genome, stripped down racing car version. It's incredibly cool. This is the organism. It doesn't look like much. It kind of looks like something that might come out of your nose on a Kleenex. But it was built using engineering, design, build, test, computer-aided design of the genome, complete synthesis of the genome, boot up in a bacterial cell. And even though this is a minimal bacterium, and it was built from scratch, 149 genes, 1/3 of the genome, is still uncharacterized. That's 149 potential PhD projects to figure out what these genes do. But this is the simplest living organism. This is why we have so much problem understanding life. We haven't done the basic homework. On the very forefront of genomics is the synthesis of the yeast genome. This was the cover of Science just a few weeks ago, March 10th, talking about synthetic yeast chromosomes. They're marching their way through them. They'll be done by the end of the year. Yeast is important because we have factories like this. This is the Tiger Brewery. They make yeast do some really awesome things, like make beer. Amyris uses the same technology to make feedstocks for manufacturing. This is a plant they have in Brazil. And a company in the Bay Area I like called Bolt Threads has taken genes from spiders and put them in yeast, and now they can make spider silk in bulk. Okay, you can buy a very expensive tie, but soon you're going to get this stuff in vast quantities. And of course, you're going to see some very, very interesting new beers come in the future. Because you can take any gene from any organism on the planet and put it into yeast. Yeast is an incredible machine. Now today I work with Autodesk. Maybe you've heard of it, maybe you haven't. It's a 35-year-old software company that makes design software. Design, simulation, visualization. Usually you use it for building cars and airplanes and buildings. But we've had a bio-nano team for the last few years. By the way, most of the people, those video games I showed you are actually made with our software. Just about any special effect in any movie is made with our software. It's a really cool company to work with because all we make in ones and zeros, and we help people create. But we've been taking some of the things we've learned in things like 3D printing and modeling, etc., and we've been moving it to the molecular. And it's been really fun. So we've made molecule viewers. Here's just a screenshot of working with a large protein. This is really hard work when you have millions of atoms. And we've also built new genetic design tools that essentially allow you to mock up genomes very quickly, make new libraries, etc. I'm not saying we're the best company in the space. There's a lot of companies that have been here for a long time, but we're learning fast, and we bring some incredible capabilities in cloud computation, visualization, simulation to the table. Maybe one day our design softwares for life will look a lot like this. Maybe not. This is just an artist's conception, but it's where I'd like to see things go. Now, I use Autodesk tools for the synthetic production of something called canine adenovirus 2. It's a weak virus, but it had never been synthesized from scratch. It's the largest synthetic virus that's ever been made. We did this last year. It was successful. We made synthetic DNA, we booted it up in cell culture, we validated it against the natural virus. It's perfect. The only reason why we haven't treated a dog yet is because it's such a weak virus, the cancer cells it needs to grow on grow faster than the virus can infect them. We're fixing that now. But this is essentially a 3D printed cancer medicine. And it's only going to get better as we start to iterate this. This was the foundation of the company we just put together a few weeks ago to be able to drive this forward, where each animal treated is essentially a whole drug development program run at a cost that's going to get very quickly the equivalent of an off-the-shelf drug. My goal is to give better cancer treatment to dogs in the short term than people get. And eventually to miniaturize and computerize this whole process and build it into a pen, a drug company, and a stick that you can just throw in your glove box in your medicine cabinet and just download that medicine of programmable nanotechnology. Since it started around 2000, it's really growing exponentially. The number, by any measurement, number of papers, number of participants. I've had the great pleasure of working with students over the last decade at a program called iGEM that was started at MIT and based on US FIRST, the International Robotics Program. And I absolutely love working with these kids. These are kids that have never been in a lab before. They've grown up digital, and they do work that is PhD worthy by working in small teams and using these incredibly powerful genetic engineering tools that large pharma companies would have just done anything for a couple of decades ago. It's a real phase shift in biotechnology. We're starting to see an industry blossom. CB Insights tallied them up a few weeks ago. Over 60 different companies working in every area you can imagine, biology touching food and drink, consumer products, healthcare, industrial chemicals, fuels, platforms. There was over $1.3 billion invested in these companies by VCs last year alone. Some of my friends at MIT that helped get this stuff rolling went on and founded a company called Ginkgo Bioworks. It's one of the most successful. It's all robotic. Another company a little more recent is Zymergen. They're focusing on materials. Again, robots. You do not want people doing liquid management. If you can see the liquids that you're pipetting, you've got 1,000,000 times too much. And these robotic labs are now going cloud. This is a company called Transcriptic. It is a programmable laboratory in a shipping container. Eventually, it'll get smaller. It is so much fun doing this work because you can actually do state-of-the-art biotech from your living room. So what's the bottleneck? The bottleneck right now is DNA synthesis. You saw how quickly sequencing dropped in price. DNA synthesis has not had that acceleration yet. It is decreasing exponentially in cost, or depending on how you look at it, increasing exponentially in price performance, but it hasn't had that wicked acceleration that we saw with sequencing as next generation technologies came about. It's coming though, very soon. The more DNA you can write, the longer you can write it, the more interesting stuff you can make. All the first generation biotech industry was limited to making very small constructs, mainly single proteins. With synthetic biology, you can start doing metabolic engineering. Now you can build pathways, not just single proteins. And what's still science fiction is being able to do large genomes, essentially write whole chromosomes or lots of chromosomes to build complex genomes like ours or the mouse. I expected we would be investing more in this clearly necessary core technology after the first genome project wrapped up in 2003. But by 2012, nothing had happened. And by 2015, still nothing had happened. And that's when I got pissed off and I shot my mouth off at a genome meeting. And I said, please, can we just start a new genome project and get the world's scientists focusing on building design tools and new synthesis technologies to go and address these important needs in genome engineering? And the result was something called GP Write, Genome Project Write, as opposed to Genome Project Read. It used to be HGP Write, Human Genome Project Write, but there was kind of a backlash. But humans are part of it. Now, I just want to say there's no synthetic babies here. Anyway, I am really, really fortunate that I have some of the most amazing co-founders of this project. George Church, who was just recently voted to the Times 100 Most Influential People, the leader of the Yeast Genome Project, Jeff Buca, and Nancy Kelly, who has been a force of nature putting together big institutions like the New York Genome Center. Our next meeting is just next week. If any of you are interested in this stuff, register now. There's probably only a couple of spots left, but this is where you need to be if you're interested in building genomes. And again, there will be no synthetic babies. This is just being able to write large, complex genomes. And these meetings bring everyone together, the ethicists, they bring together the scientists, they bring together the policy folks, they bring together the funders. This is not about any single project. You may have seen this last week. It could get a little weird in the future. If you don't want to go to New York, go to Singapore. SB7, Synthetic Biology 7. The first one was in 2003, is up and running in Singapore this summer. This brings together some of the most amazing people in science and technology. Registration is still open. Do consider it. Here's what I know to my very core. Silicon's done great things for the world. Really great things. And will continue to do great things. But programming carbon-based computing systems is going to get even more exciting. And the difference is, it's because whatever we make when we program carbon, we become the parents of. That's a whole new level of responsibility. And it means that we have to rethink just about everything that we're doing in biotech, in finance, in regulation, in business. It's all got to restart because this is such a powerful and universal technology. You'll hear from Mark Goodman later today. Mark Goodman is one of my favorite people because he is so paranoid that the world is just going to blow up. But when we get together, we have a few shots. Well, he doesn't drink, but when I have a few shots, weird things happen, like articles about biohacking and hacking the president. I'm on some watch list somewhere. But we really need to think about biosecurity in a new way, because right now the biodetection systems for most pathogens are, it's you and me showing up at a hospital bleeding out of an orifice. George, we can do high throughput robotic testing. If we're working with model cells and then moving into single cell testing, you start generating a feedback loop with real living systems that can feed into that model, and that accelerates the whole process. So I think we get really good really fast when we just learn how to work together and aggregate our knowledge. Science is incredibly fractured. Companies we expect to fracture because they need IP and all that stuff. But academics are really fractured too. Institutions are really fractured. Most genetic data can't move across state lines or out of countries. And so we're finding that this is really hard to do. So we need new paradigms. One more question and then I think we wrap up. Sorry, see me afterwards if you have any questions.
So Elon Musk is talking about Neuralink. My question is, do you think this will happen with a plant, or do you think there is a possibility that you basically can biologically program ourselves, yourself in the future to connect with the internet and so on?
That's a great question. The most exciting area in general is the intersection of electronic devices with biological devices. Whether that's the nanopore sequencer, whether it's a new type of DNA synthesizer, whether it's a sensor, or whether it's Neuralink, that interface is becoming really important. Most people don't realize living cells are electrical machines too. And you can receive signals, just think of getting an EKG or an EEG, but you can also inject signals that changes the behavior of the biology. Think more like electroshock. But Neuralink is a really neat idea, but DARPA has been thinking of programs for years as well, where they want to see essentially a cell phone the size of a mitochondrion in every cell. Now, I don't know how quickly we'll get there, but imagine just, you have a cell in your pancreas that calls you up or texts you and goes, I think I'm going cancerous. And you go, well, you should delete yourself, because the Internet of Things can apply to the 50 trillion cells in our bodies too. There's a movie out called Morgan. I saw it on the plane. It's kind of the realization of some of this technology, granted in a weaponized way. But you might want to watch it. It's kind of cool. But I really do believe that the interface of biology and semiconductors is really an exciting area and will continue to be fruitful for many, many, many engineering projects and PhD